to invite Dr. Mishra, now from Warsaw, previously from India, and after that, Dr. Mishra will move to Norway, and he will present us the talk with the title, The Role of Indirect Prey Taxes and Interference Among Predators in Pattern Formation. Please.
Thank you very much for the introduction and for saying something about me. So here in this talk, I will discuss about the mechanism of indirect prey taxes during the interaction of prey-predator. So especially there are especially two types of taxes. One is direct prey taxes, and another is known as the indirect prey taxes. So there are a lot of papers, there are a lot of studies which they have shown that this direct prey taxes always gives rise to promotes stability in predator-prey relationships. But here in this paper we tried to examine the role of indirect prey taxes. What happens during the interaction between prey and predator. So it is very important to find biological factor that may lead to formation of patches in the distribution of species. So in this paper, we've built a simple prey-predator model that describes the consumption of predator and which has some random dispersion, and also which searches for food by moving towards the gradient of some chemical released by prey. So this mechanism of direct taxes towards some chemical of prey is known as indirect prey taxes. If any prey visualize some prey, it detects some prey. If any predator detects some prey and it make direct movement towards the prey population, then it is known as the direct prey taxes. But instead of making direct towards the prey population, making movement towards the gradient of the chemical released by the prey is known as the indirect prey taxes. So in this model, we assume predator is an actively searching consumer migrating towards the gradient of a chemical released by the prey, or we can say that this is the order of the prey. While prey, here we have assumed that prey is sessile. That means the time span of prey compared to predator is very small. Therefore, we can ignore the growth and death terms in the equation of predator. So this study is especially based on the assumption that the behavioral dynamics of the predators runs on a much faster time scale than the birth or death process with a predator lifespan, being significantly longer than the prey. So here,
excuse me, excuse me, Dr. Mishra, excuse me. Do you show the model, because I see only the title slide?
Yes, sure. So I will come to the model. So this is the description. So I will come to the model. So if okay. So here are the basic assumptions. So what we have done, what is the main motive to study this model is to find the biological as well as physical factors that may lead to the formation of patches in the distribution of species. We want to the role of chemical signaling on the survival studies of the species. So how prey will behave when predator will behave when he finds some chemical of predators. Similarly, we want to build a very simple realistic model which is able to describe the emergence of space patterns corresponding to some inhomogeneous distribution of species. So we have made some basic assumptions, and so the basic assumptions are that the behavioral dynamics of predators runs on a much faster scale than its birth or death process of a predator. That means here we can ignore the birth and death of predator in its growth equation. Here we have only assumed the aggregational response of predator, which may change the prey distribution. So here we can see the model here. So here you shows the density of predator. W shows the chemical released by prey, and V is the density of prey. So here we have three equations. UTVTW shows the rate change of particular individuals with over time. So here we can see that in prey equation we have assumed that as I have here mentioned that. So we assume that predator runs on a much faster scale. So it has very long lifespan, significantly longer than the prey. So here we have not considered. You can see the first equation. This equation is equation of predator growth. So here we have not considered the birth and death process. We have only considered the aggregational response, only diffusion. So here we can see in the equation of prey. So there we assume that there is a logistic growth in prey, where lambda shows the growth rate of prey and K is the carrying capacity. So this is very well-known function, logistic growth function. And we all observe that only aggregation of prey, only the density of prey can alter the population of prey. So here we take this FU. This FU shows the rate of consumption of prey by predator. So here FU, and we assume that prey releases some chemical, which is well known in aquatic system. Prey releases some pheromones, some chemicals. It may be some sexual chemicals. These chemicals may be released from the body of any predator, or this chemical also it can be released during the encounter between prey and predator. So here we have assumed then that predator releases some chemical. Here alpha is the rate of production of chemical, and you can see this is the alpha is the rate of production of chemical, and here mu is the degradation rate of chemical. So here we have ignored the diffusion in prey. So here we have observed that the chemical of prey is spread with DW rate, and the rate of spread of predator is DU. So here we have incorporated, we have an assumption that predator. So here you can see divergence. So predator moves towards the gradient of chemical. So here this term, this nabla W shows the gradient of chemical at some particular point of 2 dimensions of a space. So here dW, delta W shows the gradient of chemical, and the predator moves toward the gradient of chemical. And here this key term shows the chemotactic sensitivity constant. So here this term shows the direct movement of predators towards the gradient of the chemical, and this equation shows the production of the chemical over time. So here this model shows the indirect prey-taxis mechanism in prey-predator relationship. So here we have seen that there is no growth term in predator and prey. There is no growth term in the equation of the predator, and only this FU functional response can alter the population density of prey. So here we have taken this. So now here we have taken a unimodal type functional response. So here, now I would like to show how we have derived this equation. So we just take simple Beddington, well-known Beddington de Angelis functional response. Here you can see this functional response assumes both the density of predator and also density of prey. So to derive this functional response, which accounts only the density of predator, we have assumed that the handling time of prey is 0, like it is already mentioned in the Halling 1 functional response. So we have assumed that the handling type of prey is 0. So then, and we assume that this term BTW is equals to this quantity, and then when we put this value here, then we can find this equation. So here we can find a functional response VFU equal to this one. This is AVW plus M minus 1 UC M power M into U to the power M. Please remember that here U counts for the density of predator and V counts the density of prey. So here notice that if M is greater than 1 and U is greater than 1, so FU is a unimodal functional response. That means it attains its maxima at some point, say U equal to UC. It grows until U to UC, and then starts decreasing after this point UC. So if similar functional response is also proposed in the aspect of prey when there is mono-Hurden functional response of type 4. So Halling type 4 has the similar mechanism when there is only prey, there is no predator. So here we have this functional response shows that aggregation of predators gives rise to decrease in the competition, decrease the competition. That means if there are too many predators, then the competition may then this density, then that means the mortality of predator will decrease. So here in this equation, so if there is too many predators, then we will and the predators crossed the UC this threshold value, then this mortality of predator will automatically start decreasing. So here, so this is the unimodal functional response. So this now, so this is visible to hello.
Excuse me, could you please explain the meaning of this condition? Why consumption of prey decreases when the density of predator increases?
So imagine the situation when there are too many predators and they are interfering with each other. So if there are too many predators and they fight with each other, then definitely they will handle low number of predators. So in certain case there is a possibility that this functional response has a minimum value. So in this case, then the mortality of prey will automatically decrease.
Thank you.
So it is clear. So if there are too many predators, then they will interfere with each other and they will fight with each other. Then definitely they will get low food. They will spend more time in fighting with each other. So this is some kind of interference. This is strong interference between each other. Yeah,
I understand what you say. I mean, well, it can be discussed. I think that this condition can be discussed because the predators begin to fight each other when there is no enough food. So first they eat food and then they fight. but anyway, we will discuss it later if we have time. Please continue.
Okay. Thank you. So here, we made some modifications and we non-dimensionalized the system. And after non-dimensionalized systems, we get following system equation, which accounts the indirect paradoxes, and we can see this function of this form where fu can be defined as AU upon divides by some 1 plus Cm plus Um. So imagine if M is 1, then this is some kind of Halling 2 response. And if M is 2, here M is 2, then we can say that this is some kind of model function where it increases until for some value of U, and then it starts decreasing. So this is some kind of Halling 4 functional response, which shows similar behavior when there are too many preys. So here, and it can be easily shown that this function f attains its maxima at u equal to 1. So here this it can be seen that this equation has ut and vt contains diffusion terms, and this equation of prey does not contain any diffusion term. So such kind of quasi-linear PD are known as the degenerate PDs. So there are some theories, semi-linear theories. So with the help of this theory, we can prove the existence of the solution of such systems. So now I will start. So here we can see that the model is a couples quasi-linear parabolic system with an ODE and may be classified as a degenerate parabolic system. And this is defined in we define a simple Sobolev space of this kind here, and it has been shown that for a positive initial data, if we take some positive initial data, then there may exist a positive solution of this PD. So with the help of this system, so with the help of very classical comparison method, we are able to show the existence of solution, and it can be found in the paper. So here I am not going to discuss what we have done here to show the existence. So this is presented in many papers, and there are many papers they have shown the existence of the solutions. So now I will go to the linearization. So in order to
sorry, excuse me, I have still a short question. Though you don't discuss the proof of the existence, but I have a short question concerning this taxis or cross diffusion term, because the existence of solutions is quite simple, or maybe even very simple in the case without cross diffusion, but sometimes it becomes much more involved in the case of the presence of this cross diffusion term. So in this case, this cross diffusion, does it make it more complex? No,
it is not complex. It is already shown in many papers. So this is not very complex. There are many papers where they have even there is a paper published by Tello and Wroszek. So they have shown the existence of solutions.
Okay, thank you.
So this is not very tough, and our readers can find its proof. Okay, so now I will go to actually our main motto is to show the pattern formation mechanism of pattern formation. So we will discuss here the process of linearization. So we disturb the system. We disturb the system and we examine what happens on the stability. So here we have this system. So so here we can see that we have this system. So in order to find steady states, we put this system into this form, and then we found that from this equation we can examine, we can calculate the expression of V. So here we can see the expression of V can be written as this form here, alpha into 1 minus fu divides lambda. So we can put this equation here in this equation. So we will find the only two equation here, this equation and this equation. So here the study of this solution may be reduced to, so we reduced this three equation into two equations, and there are many papers who shows that they have shown that the dynamics of this system and dynamics of this reduced system, they are similar. Even in later stage, we will also show that the dynamics of this system and dynamics of this system are same, and with the help of numerical simulation. So here we can calculate this value of V. So here you can see the value of V by putting this equation equal to 0. So V can be calculated as V equal to 1 minus FU by lambda. If we put this value here in this equation, then we will find these two equations. So these two are, so here equation 7. So with the homogeneous Newman boundary conditions. So here we assume that the average concentration of parameter over the domain is constant. So here m is a constant, you can see. So we make an assumption that average concentration of the parameter over the whole domain is positive and it is a constant value. So with this assumption, now we are able to show that the bounds for W, I mean the population bounds for W and U. U counts the density of predators and W counts the concentration of chemical. We are able to show that there exists some priority bounds. So here, with the help of standard comparison lemma, it is very easy to show the bounds. So here we can see that WT is bounded by this quantity, alpha by U, 1 minus F bar by lambda, and alpha by mu, and the density of parameter is bounded by these two values. So here this shows that we have a classical solution. We can get a classical solution because we are able to show the bounds for the parameter and the chemical. So these bounds can be very easily can be calculated with the help of very standard comparison lemma. So now we go to the to examine the impact of perturbation on the steady states. So here we have this eigenvalue problem. So we have some such eigenfunctions defined on this Newman boundary condition. So let we assume that lambda n and wn are the sequence of eigenvalues, and their corresponding normalized eigenfunctions are defined as this. So then if we substitute these values, so this is very standard, I think the linearization is standard. So if we substitute this value here, so we will get we obtain this matrix, this Jacobian matrix, and here we can see that. So from this matrix we can easily calculate the eigenvalues. So from this matrix we have calculated the eigenvalues. So here we can see the following eigenvalues. So here from this eigenvalue, here two eigenvalues are negative, so it's okay. So here we can see that from this eigenvalue can here. So here if we especially look at this function fu. So this function fu is unimoidal, and we take its if we take its derivative, so it will be positive. So this eigenvalue can be positive if this key exceeds some value. So here f dash u is positive if we find the derivative of this function fu here. So fu is defined is somewhere here. So here fu is defined is here. So if we take derivative of this fu with respect to u, so then this fu will be positive. This fu will be negative, and then this term goes for positive. So then we can find a key, this value key, so that this whole eigenvalue may be positive. So here we calculated this critical value of key, say key n star. So if this key n star, if this key is greater than key n star, then definitely these eigenvalues will be positive, and then instability may occur. And if this key is less than this key n star value, then definitely this eigenvalue will be always negative, and there is always a homogeneous steady state, and there is no pattern formation is not possible. So here, so we are able to calculate this critical value of key, so that we can observe some patterns. But please remember that if we have this direct axis, so here we have imagined the indirect rate axis, but in case of direct rate axis, it has been shown in many papers that the steady state remains stable always. So that means if there is also tearing stability in such preparator models, and we have direct axis, we incorporate direct axis over tearing stability, then it may destabilize the steady states. So here we have seen just different mechanism. In other sense, if there is a direct axis, then steady state will always stable, and there are no pattern formation. But if there is,
excuse me, I have a question for you, because here you have a possible instability, not because you have the indirect axis, but because you assume that the function f can be decreasing.
Yeah. Sure. So
if f is increasing, so in this case of indirect taxes, you don't have instability neither.
Actually, it is already shown by Italo and Rosek. So they have studied a paper where this function f uses of monode type. So Halling 1, Halling 2, Halling 3 function has no impact on this eigenvalue. So if we have monode function, then always there is stability, and it always promotes stability too. So if there is monode function, then always even the direct axis and also this indirect axis promotes stability. But here in this case, if we have this function fu, so if there is aggregation of predators, then only it is possible that in case of indirect rate axis, there may have instability. So here we are able to find this key n. So if steady states become stable, unstable if this key value is greater than key n star, and it is stable if this key n value is less than key n star. So now we are here. Now we will show our results with the help of some numerical simulations. So to this end, we have fixed some parameter values. These are taken from different biological papers. So we tried to find some real biological parameters, and we calculated that we assume that U bar, which shows the average concentration of predator, is taken as 1.1, and we calculated that the concentration, that the steady state of W is calculated as 1.557. So we use MATLAB PDP tools to simulate our model in 1D case, and we apply free FEM++ package to simulate our model in two-dimensional space. So we have first we are going to show the simulation results obtained in 1D case. So please remember that in simple preparator models without this cross-diffusion terms, there are plenty of papers. They have shown the Turing-like instabilities, and they have no such spiky solutions. So here we have tried to find different types of spiky solutions, and we have especially examined the role of initial data on the solutions. So here first we take initial data, so which shows the at node 2. So if we take initial data and we run the simulation, so it can be seen that after some moment, the steady state goes for state becomes homogeneous, and it converges to its steady state. So if here you can see, if the key is less than key star, so initial perturbation converges to its homogeneous steady state. So
sorry, excuse me. So the figures are a little bit small, so I think that it's better to explain that there is x-axis, right, and t-axis, right? Because it's really small, so we cannot see. Yes, explain please what are the axes.
So the x-axis shows the distance. So this is the space y-axis shows the time. This is the time, and this is the density of the preparator, and this is the density of the chemical. So here you can say, so this shows the density of chemical. So this is the time, the axis of time. This is the axis for the space. So here we have, if we make a small perturbation, initially there's a perturbation, and after some time it converges to its steady state. So here we can see that it converges to steady state. So if key is less than key star, then a small perturbation converges to steady state. So if key is greater than key star, so we can see here, we can see that this perturbation is rippling for key greater than key star. So then we can see the instability, and it does not there is perturbation. If there is a small perturbation, and it converges to some different solution, so it does not converges to the constant steady state. So there may have some there are some patterns depending upon this key greater than key star. So now here, now we have shown the impact of this key on the steady states.
Sorry, excuse me, I have another question. But you consider here the simulations for 2 equations model or for 3 equations model?
No, this simulation is for reduced system. So this solution is for
but you obtain this reduced system for the stationary solution without time dependence. But here you consider with time dependence. So you cannot reduce 3 equations to 2 equations for the time dependent system.
So actually, if we calculate here, so from here you can see here, if we find, so if we put from here we can calculate this v, and if we can put here in this equation, so this equation is reduced into 2 systems, and they both have the similar structure.
Of course, I understand that if you consider the stationary case, you reduce 3 equations to 2 equations. In the case if you don't consider if time derivative, you put time derivative 0, then you have the stationary case, then you can reduce 3 equations to 2 equations. But you show, as far as I understand, you show numerical simulations for the time-dependent case. Could you please show the time-dependent system in the beginning, 3 equations with time derivative? Then you cannot express v from the last equation. So probably you consider kind of maybe a quasi-stationary approximation for the pre-density, right? Then okay, continue, please.
Okay. So, for the next simulation, we have taken this initial data for the preparator and chemical, and we run simulations. And for this figure A shows the simulation for the chemotactic sensitivity key equal to 7.5, and figure B shows the simulations for the chemotactic sensitivity key equal to say 10. So here we can say that this simulation is very similar to initial data, and they are not very sharp. So they are not very sharp, and if we increase this value key, so we can see that spiky solution, this boundary spiky solution may occur. So here we want to show that if this key is much bigger than the threshold value of key star, so then a very spiky solutions can be occur. So here in the next simulation. So here now in next simulation. So in this simulation we try to show different types of spiky solutions. So in the first solution we can see a solution of V shape. V shape, and here you can see maybe it is visible. So this is visible to you. So this solution is obtained for key equal to 8. So we have taken this initial data, and it is for key equal to 8. We have solution of this kind V shape, and if we increase this value, and take a snapshot of solution at k equal to 15, then we can see the solution of V shape, and we can see that there are two spiky boundary solutions. So we have two boundary solutions, boundary spiky, double boundary spiky solution. And if we make small further change in this amplitude equation, this S, and we take this minus, so we can see that there are different kind of structure appears for the solution. So here we have this space which is in a model shape. And similarly, a spiky solution in the middle of this middle of the domain is observed when key is too high. It is for key equal to 8, and this is observed for key equal to 15. So here both the figures, so we can interpret the figures that even if in the patterns there is a bigger effect of this parameter key, and also it depends of initial data which we have chosen. So the patterns, pattern 1D.
I have a short question or comment. But in fact, in both cases, you can extend this solution by periodicity to a larger interval, and then these two solutions are in fact it is the same solution shifted in space.
Yeah, so they are the same solution, only we have changed the yes. So this is shifted in space. One is in the corners and one is in the middle.
Right. Thanks. Continue, please.
So now we use free FEM tool to show simulation in two-dimensional space. So the figure A corresponds to the initial data of this initial data, where we have such kind of initial data, and we have taken U which corresponds to the initial data for the predator, and W0 corresponds for the initial data for the prey. And for this initial data, we observe that there is a very huge gradient of prey, and we can see one spiky solution which is boundary spiky at 1, 1. So we can see a boundary spike at 1, 1. So we can see here this spiky solution is very similar to the initial data. So similarly, for different initial data, for the figure B, we have chosen this initial data UR plus this one, and for this initial data, we have observed two spikes, both in the corners. So here one is in the corner 1, 1, and another is at the corner 2, 2. So if for different initial data, we have two different kind of solutions of our system. So this reflects that the patterns of such system depends upon the initial data. So biologically, we can say that the patterns in the space depends upon the initial distribution of prey and predator and the chemical. So in the next figure, so we have again changed some initial data. So we have such initial data of this kind. So here U0 is taken as this one, and W0 is taken in this form. And for this, we can see that there are two spikes, and they are in the boundary spikes, and one is at 1, 0, and another is at 1. So here again we have some different structure of pattern. So similarly, again we have changed some initial data, and we have 3 spikes. One is in the boundary, and 2 is at the corners. So now we have examined the impact of this patterns, the impact of domain size on the pattern formation. So maybe this figure is very small, so I think I should. So. So here you can see, so this is x-axis. So now this simulation are present in two-dimensional domain. So this this shows x, this shows y, and this shows the prey population, I mean the density of predators. So similarly, so the first figure is shown for the unit domain. So we have on unit domain. So in the unit domain, we have taken some initial data and we run the program. So we observe that there is only 2 spikes in the corner. So we can see 2 spikes on the corner, and for the same initial data, and for the same initial data and for same parameters, we have just increased the domain size, and we have domain size of 5 cross 0 to 5 square. So we have in this case we have many interior and boundary spikes. So here we have observed that if we have increased the domain size, then the number of spikes also increases. So similarly, in the next, we have taken domain size of 0 to 15, 0 to 15, 0 to 15, and then we can see many of spikes in the domain. So this figure concludes that we can find more patches of prey, or we can see many patches of prey, or we can say many spikes in space if domain size is big. So this result shows that the domain size has a larger effect on the patterns formation. So this is the snapshot. So this is the snapshot taken at time t equal to 1000, and at that time we have observed that there are patterns, there are spot patterns, and there are spot patterns here. So usually such spot patterns are not possible in certain preparator models, but here in the case of indirect axis we obtained some patterns. So these patterns are not already obtained in when there is a taxis mechanism. So but in this indirect axis mechanism there are some spot patterns. So such spot patterns can be also observed in the Turing stability. So here, so this finally our result concludes that. So now there is one more figure here. So now this figure is shown for our original system, and for the original system we have shown that if we have some key greater than key star, we have some value of key which is much bigger than its threshold value, then there is possibility of patterns in original systems too. And we have observed that the both system, original system and reduced system, has similar behavior. So finally, this study suggests that if there is indirect rate-axis, then there may have patterns. Instead, if we consider direct rate-axis, then there is no possibility of such patterns, which is already shown in many preparator models. And even if there is Turing stability, then this direct axis will homogenize the steady state. But if there is indirect rate-axis, if we consider indirect rate-axis, and also we consider this aggregation of predator, that means if there is a functional response which only considers the interference among predators, then there is a possibility of patterns in such preparator models. So this is the final conclusion of our study.
Thank you very much for this very interesting presentation. And please, if there are questions, we have now time to discuss, please. Okay, while waiting for other questions from the audience, I'd like to ask you if you can return to the slide with two-dimensional simulations, just 2 or 3 slides before. Yeah. For example, what's this one? This one is good, right?
This one or this one?
This one, yes. Slide number. Well, I don't see the number. Anyway, this one. So you have this here several different types of solutions with two peaks, with 3 peaks. Or on the previous slide, it was with 1 peak. So when you do the linear stability analysis and you find the eigenfunctions, so do you have eigenfunctions, for example, with 3 peaks like shown here?
So eigenfunctions.
In fact, I will explain why I asked this question. Because you see that what you say is that you take different initial conditions and you can obtain different patterns. Problems like that, it is possible that you have very slow time evolution of the solution. So if you wait a very long time, so your solution with 3 spikes, maybe it will become the solution with 2 spikes or with 1 spike. So maybe it's not really a stationary solution. This is my question. And one of the ways to verify that is to look at the eigenfunctions. If you have an eigenfunction, for example, with 3 spikes, it will be an argument to say that when you have a bifurcation of solution, so you can have a bifurcation of solution with 3 spikes. So I repeat my question. Do you observe eigenfunctions, in fact, several different eigenfunctions for the same eigenvalue with different number of spikes or peaks?
So we have eigenfunctions. So I can show you the equation of that eigenfunction. So I think I have put in here in the simulation. Please let me open the slide. So here you can see. So this is u bar key n.
Yes. So this u bar is what?
This is u bar. This is the average concentration of the predator. So this is m. If you remember.
No, sorry. Sorry, but I don't see the eigenfunction, or I don't understand what this formula means. So u bar is a stationary solution, in fact.
Yeah.
You linearize about the stationary solution, find the eigenvalues, and define the eigenfunctions. If you don't, okay, anyway, if you don't have it here or you don't remember, never mind. It's then I will ask another question. When you have, again, can you return please to the slide with these 2-dimensional simulations where you have the several, oh, okay, this one, yeah, this one, yes, no, the next one. The next one.
This one? Yeah.
Yeah, this one. As far as I remember, you mentioned the initial condition here is the same as in the previous case, but the domain is different. When you say that the initial condition is the same, do you mean that if you consider this larger domain, in the initial condition, how many spikes you have in the initial condition? Only 1 spike and then you obtain the solution with spikes, or you have the same many spikes in the initial condition?
No, for this we have 1 spike. Similarly, if here too we have 1 spike and here too we have 1 spike. So initial condition means we have similar number of spikes initially.
Similar. No, could you repeat please? Similar
number of spikes initially.
Similar number of spikes. For example, if you take at the bottom figure C, so in the initial condition you have 1 spike or 20, I don't know, 20 or 30 spikes in the initial condition.
So like here. So here. So here. So you are talking about this figure. You are talking about this figure. So here we have 2 spikes. So initially we have 2 spikes. So these 2 spikes emerges and we have such solution. So then here also we have 2 spikes, and here also we have 2 spikes, and here also we have 2 spikes initially.
Okay. So initially you have, in the initial condition, you have, like, for example, 2 spikes, but then you observe emergence of other spikes in the solution, right?
Yeah, sure. Yeah, sure. This is what I want to say.
Okay, thank you very much. Okay, please, other questions.
May I ask a question?
Yes, please, of course.
In your model, you have no diffusion in pre-equation, right?
Yeah.
Is it a crucial assumption for?
Actually, you can see in the aquatic ecosystem. So there are many phytoplanktons. So like like flowers. So so they don't move. They don't diffuse. So this is taken from the aquatic ecosystem. So this is not very standard, but few examples are there in ecology.
But from the
what will happen if you will have some diffusion?
Sorry.
From the mathematical point of view, it will change completely the it will change completely the system because because you cannot express v from the third equation.
Yeah, sure. Then everything will change.
All the solutions were shown for this simplified system, right?
Mm-hmm. Yeah. So here is the model.
But it is possible still, but it is still possible to do linear stability analysis even for 3 equations. So from this point of view, it is possible, of course, to consider the case with diffusion.
Yeah. So yeah, sure.
Actually, more than 20 years ago, we started with my colleagues to study a very similar system, but we had even much simpler trophic function here. We had a linear Lotka-Volterra trophic function, but the same system and with diffusion of prey.
Okay.
And we had also conditions for emergence of heterogeneous but dynamic, non-stationary, very complex solutions. You're right.
Did you also consider taxes of predator?
So if there are monod function here, if there is this function is monod, then there is no instability. I mean, then we cannot find patterns. So patterns is
no, you are not correct. I believe. If you here have just a linear function, for example, a multiplied by u by v, then you have everything in this model.
No. Actually, maybe there is a paper published by Tallo and
I know, but you see, you have very special case of predator interference here.
Yeah, sure.
Without this interference, when you have just normal functional response of predator, even without saturation of predators, you will have very interesting solution with this model, heterogeneous. You can find some of my older papers in ResearchGate, for example. Just to see.
Yeah, sure. I will try to find
the papers. Very similar system, but not with this indirect taxes, but with you see, if you will differentiate the second equation here, you will have equation for taxis velocity.
Yeah.
That's what we had in our model.
Okay.
And but much simpler expression for f.
I will try to find your paper. Maybe it is interesting to read.
Yeah.
Thank you for this interesting comment. Please, other questions? Okay, if no questions, then let's thank again our speaker, Dr. Mishra, for this very interesting presentation. Thank you very much. And then for the listeners of our seminar, we will meet. Anastasia, could you recall us, please, when we have in 2 weeks we have our next seminar, right?
At 25 of February, we have 2 talks. The one from 3 p.m. until 4 p.m. This is Elena Kurbanovna Shavarova with talk about pressure.
Blood pressure
measurement. And the next talk will be from 4:30 p.m. till 5:30 p.m. And it will be Sergei Vladimirovich Bernikov.
Okay, very good. So all our listeners are kindly invited to the next seminar in 2 weeks. And now we have finished with the seminars. And for those who are interested, we meet half an hour later for mini conference called Round Table, and its members of our center will present short talks basically for students about different biomedical problems. But these presentations, I believe, will be in Russian, right?
Okay. So maybe this is not convenient for me.
So for those who know Russian, or for those who want to learn it.
First I have to learn Russian.
Okay, thank you very much. Thank you
very much for inviting me for the talk.
Oh, it's a pleasure. Nice. It's a pleasure.
Yes. It was nice meeting with you here, and really I enjoyed it. Thank you very much.
Okay, thanks again. Bye to everybody. See you in 2 weeks.
Yeah.
Thanks.
Thank you. Bye to everyone. Bye.
Yes.
Thank you. Goodbye.
Goodbye.